Welcome back. We are on section 3 of chapter 1, and the title of this section is Methods of Science. And the main idea, please write it down, biologists use specific methods when conducting research. So, science. Ask a question. Scientific inquiry begins with observation. And scientific inquiry involves asking questions and processing information from a variety of reliable sources. So what is the scientific method? Well, scientific inquiry begins with an observation, a direct method of gathering information in an orderly way. The process of combining what you know with what you have learned to draw logical conclusions is called inferring. The conclusions themselves are called inferences. The methods scientists use to gather data and answer questions are referred to as scientific methods. A hypothesis is a testable explanation of a situation. Well, what does that mean, testable explanation? Well, I'll tell you. You cannot test what someone is thinking. You just can't. You cannot test exactly the words that are coming into their head. Now, can you test if there's an emotional response to certain types of words? Sure you can. But you cannot test their actual words, the vocabulary that's going through their head. So in order for a hypothesis to be a true hypothesis, it first must be testable. When a hypothesis is supported by data from multiple investigations, the hypothesis is usually considered valid and is accepted by the scientific community. If the hypothesis is not supported by scientific investigations, the hypothesis is revised and additional investigations are conducted. And if I could add another bullet point, I would add there, investigations and revisions continue until there is a conclusion. Sometimes there's simply not a conclusion. So, collecting the data from controlled experiments. When a scientist conducts an experiment, they investigate a phenomenon in a controlled setting to test a hypothesis. The control group in an experiment is the group used for comparison. An experimental group is a group exposed to the factor being tested. Before I go any further, this part right here, exposed to the factor being tested. Let's talk about that. We're talking about variables here, okay? So experiment design. During a controlled experiment, only one factor can change at a time. Well, what do we mean by factors? Independent variables. These are the tested factors that might affect the outcome of an experiment. And the dependent variable results from or depends on the changes to the independent variable. So let me stop for a second. An independent variable. If I wanted to see if the medicine was working for a migraine, I would give people a certain type of medication, and I would see if they reacted to it or not. The independent variable in this case would be the medication I gave them, or the amount, I should say, of medication they were given. And the dependent variable is the result or the thing that I can measure. So next to dependent variable, add something I measure. And next to independent variable, please add something I change or is within my control. Again, independent variable, something that I can change or is within my control. And a dependent variable is a result of that change. It's the thing that you can measure. A constant is a factor that remains fixed during an experiment while the independent and dependent variables change. So, let's say for a constant, off in a separate area of this test or of the room, I have people who are having headaches, migraines, and I give them medicine. But it's really a sugar pill. It's not really medicine. They just think it is. and then I compare their results to the results from my dependent variable. Data gathering. As scientists test their hypothesis, they gather data, information gained from observations. There's two different types of data that I'm going to be asking you for all year long. And for many of you and for a lot of our experiments, it's either going to make or break your grade. And those two things are quantitative and qualitative data. Quantitative data can be collected as numbers, such as measurements of time, temperature, length, mass. You get the point. Quantitative, next to quantitative, I want you to write quantity. That's what you're doing. You're measuring a quantity. Qualitative data are descriptions of what our senses detect. So, what am I smelling? What am I seeing? What am I hearing? And please, in the science lab, not what I'm tasting. Data is organized in notebooks and tables. Investigations. Not all scientific inquiry is based on controlled experiments. Investigations rely on observation and collection of data rather than manipulating variables. For example, the metric system. It uses units with divisions that are powers of 10. SI units, International System of Units, are commonly used in science for consistency and ease of communication. So instead of using inches, we use centimeters, millimeters, liters, etc. Analyzing the data. I'm going to be giving you a lot of these type of sheets during the day when we're working with each other in class, and you're going to need to analyze them. And for peer review, you're going to be looking at your peer data, and you're going to be analyzing and grading them on that. So after data collection, scientists use analyses to ask questions such as, has my hypothesis been supported? Or, are more data needed? Patterns in data can be displayed in tables and graphs. Data analysis determines if the hypothesis is supported or rejected. Reporting conclusions. Guys, I cannot tell you how many people do not do well on this. They give me, yeah, it supported my hypothesis because, and they kind of sort of restate the hypothesis, and they might throw me in a little nugget of observation. And no, I want a true report of your conclusions. Before a scientist can publish their findings, it must be peer reviewed. And if the reviewers agree on the merit of a paper, it is published in a scientific journal. And other scientists use published papers to evaluate and develop their own research. Think about it for a minute. If you're only giving 1 to 3 sentences, what is there to peer review? Not a lot. You need to give the reviewer lots of information from your data, your observations, everything that you took away from your lab. Please make sure you do that. Student scientific inquiry. As a biology student, you will have the opportunity to participate and design your own investigations. Lab safety practices are designed to keep you, your classmates, and your teacher from harm during experiments. And lab safety sheets will inform you of any hazards present in an activity. So basically that means by the middle of the year, and especially by the end of the year, you're going to be like, oh yeah, I know what I'm doing. And you're going to kind of skip that part, and you're not going to read what the hazards are. Every time, read the hazards. If there ends up being lots of accidents or a lot of people not really following safety rules, labs are suspended. And this happened last year. I don't want to see your class have a lab suspended for the school year. Please make sure that you take precautions and you take lab safety seriously. And for your pop quiz, make sure you can answer these essential questions. What are the differences between an observation and an inference? What are the differences among a control, independent variable, dependent variable, and how do those three things work together? What are the scientific methods a biologist uses for research? And why are the metric system and SI important? Take a note of your vocabulary, which we've already reviewed in this session, and be prepared for the test coming up soon. See you tomorrow.